Thanks everyone for coming out. So I want to start talking about inventions. It's one of the core concepts that kind of drives the way we think about things. And if you look through history, there have been new tools that have changed the trajectory of humanity. And the idea that you can do something like that is really fascinating. And I think it's particularly interesting when you look at therapeutics, and we just heard the wonders of the microscope. Looking at that and some of the things that came out of that of understanding of cell biology and germ theory and development of antibiotics, those have had a meaningful impact. If you look over like the last 150 years, human life expectancy has about doubled, which is amazing, but that has really slowed. Development of new therapeutics is very problematic. Right now it takes on average about 20 years and $2 billion to get a single drug to market. And there are about a 90% failure rate for drugs entering clinical trial to make it out. So we focus heavily on trying to find a tool that can help to reduce the time, cost, and failure in drug development. So today I'm going to tell you a little bit about the drug development process in general, give you an overview of the new tool that we've developed, and then an example of how we've used that to come up with a new therapeutic for Parkinson's. So we focus on neurodegeneration, which is a difficult space. Not a lot of people are, not a lot of progress in therapeutics for this area. If you look at some of the others, the changes of cause of death over the last 15 years or so, good progress in cancers, stroke, HIV. Neuro's headed the wrong direction. These stats get worse year over year, so there's a huge unmet need. Maybe more importantly, though, for what we're doing, it's a complex area. The cellular biology, the structures in these systems are very complex, and it makes it difficult to use other approaches. Some of the computational methods to understand these neurons and the biology of disease have been challenging, so we thought a new approach would be especially useful here. So if we're trying to develop therapeutics, I think it's useful to run through what defines a good therapeutic, what's going to make a drug that works. You need to have some molecule that effectively engages a target, where that target engagement reverses the disease progression, some phenotype that you're trying to change. Has to do that safely for a disease where there's actually a need. And then you have to be able to find the patients to give that drug to. And there's been some good progress in a lot of these, but particularly around finding the right targets and finding the right patients to give the drugs to. Those are pain points that we've specifically tried to address. And if you look, there was a study published in Nature a few years ago looking at the reasons for failure of clinical trials, and about half of them fail for efficacy, meaning they got through safety trials, all of the preclinical data looked good, they knew their drug hit their target, but it didn't affect the progression of disease as expected. So if that's happened, that really comes down to either your target was wrong, you picked the wrong thing to modulate, or you didn't give the drug to the right patients. So being able to identify the underlying mechanisms of disease, to understand why your target is the right target, and to be able to stratify your patients to know who you're going to give this drug to are going to be huge factors to try to get to more successful clinical trials. So this is an exciting time to play in the space of computational approaches to biology for two main reasons. First, there's a ton of data available. We work with a variety of data sources. I'm not going to go into them all here, but having the ability to integrate data from these places lets us do cool things. And then preaching to the choir, Nvidia GPUs have opened up new doors. Even just a few years ago, you would have had to kind of be in the inner circle with a handful of supercomputer labs around the world to do meaningful high-performance computing. And now I can provision a cluster of Nvidia GPUs on Azure in minutes, run high-performance simulations, and tear it all down when I'm done. But all of that data has another big problem. Different groups name things differently. The data from is from all these disparate sources is difficult to bring together. So there's a big challenge to unify that data into one platform where we can work with it and do analysis. Once you get it into one place, though, then you can do really cool analytics, simulation, which I'm going to get into a little bit more, or artificial intelligence approaches that a lot of people are working on. Another big challenge is knowing what data is good, the need to curate and validate the data that you're going to work with. If you look through biological publications, you're going to come across discrepancies, contradictory findings. Biology is constantly changing, knowledge is getting better over time. So this is where we think it's really important to integrate with bench biologists. You have to have scientists that really understand the biology working with the system, tightly integrated into your work, or you're going to end up running simulations of bad data, getting bad results. So with that, I'm going to switch tracks to the actual platform. This is the kind of an overview of the simulation system that we've built. To facilitate getting those biologists' hands on the system, we started with a graphical interface that allows the users to build models, run simulations, visualize those simulations, do analysis of the data, all without writing any code. Built that using the Unity 3D game engine. Had some good benefits there. A pretty standard REST service behind that to get to the data and do analysis. This also allows us to do all of the same functionality from scripting sources, Python, R, and whatnot for the power users. And then the real heart of the system is a custom simulation engine, which is built on CUDA and allows us to do a remote simulation at scales that don't work on a single, on a local laptop. At our peak scale when we're running, our cluster is about 100,000 GPU cores, which is kind of cool to be able to get that and not have to keep it going. Takes us about 15 minutes to bring up the cluster, run our simulations, tear it back down, and not pay for it when we're done. So that's been a great benefit. So when I say simulation, I'm talking a 3D spatial temporal representation of a virtual cell. This is a screenshot from the application. You can see the cellular context, what part of a subcellular system you're looking at. Zoom in, see the biochemical entities that are within that area, which include proteins, lipids, small molecules, also larger structures of organelles, compartments within those organelles. And the complexity of this environment with localization and fission and fusion of these membrane-bound structures is highly dynamic and provides challenges that I think cause some of the differential equation-based approaches to systems of biology to come up short, where our approach closes some of those gaps. Having this virtual cell allows you to do two really important things. First, you can create a disease model by introducing genetic or genomic manipulations and measure the abnormality, see what goes wrong in your cell. Then you can add therapeutics to your sick cell and see which manipulations are going to bring that cell back to health. That's what we do. So diving a little deeper into the data that drives this, there are two main categories. The first are spatial content, represented in a structure of the extracellular space, collection of entities within that, along with the properties that make up those entities, collections of bounded containers, cells at the outside, organelles, interluminal vesicles, other compartments below that. Quantities of the entities come from some omics data, Transcriptomics, mRNA is what we usually work with because it's cleaner, more consistent. Can also work with proteomics data, anything that gives you a quantity. Additional challenges of knowing how to distribute that quantity across the cell. To do this kind of simulation, you have to know what portion of that to put in the, say, the lysosome versus the organelle, or the mitochondria versus the cytosol. So that's a bit of an extra challenge to work out. The next piece of the data are interaction rules. So you have to have a behavior for this thing to do something. This is a screenshot from Intact, one of the largest protein-protein interaction databases. And this is, will tell you that two entities interact. You have Rab3A listed under molecule A and LARC2 listed under molecule B, but that's not enough information to actually simulate or run a virtual system. So here's a simplified example of how we expand that out within our platform, where we know LARC2 has to be bound to ATP in order to participate in this interaction. And when it does, an outcome is that a particular post-translational modification on Rab3 and ADP released as a byproduct. Now, this is just one simple example of the variations there may be. Both of those entities may be on or off a membrane. They may be bound to other entities like GDP or GTP. So there may be hundreds or thousands of variations to consider for any single protein-protein interaction pair. Looking a little bit further, getting into what the simulation does, the method is basically a time-stepped particle system. There are examples for this in the Nvidia GPU gems, kind of to get started off. It's a basic time-step loop where you integrate the positions for all the entities, detect collisions, and resolve the positions. So normal physical interaction, but then within that, we evaluate the rules, like I just showed, to determine entity state changes, and then also log results for post-simulation quantitative analysis and stream positions back to the users so that the biologists can actually view a running simulation that's happening on the cluster at real time, which doing that without blocking the parallelism is a challenge I won't get into today. Looking at some of the math underlying the system, it's Newtonian physics, Verlet integration, and perfectly elastic collisions. So a simple rigid body type method. And then with any new system, one of the first questions is, does it work? How do you know that it works? So our metric is comparing to biological results. We're trying to simulate biology. What do we see in an animal model when you look at the same manipulation? So this is an analysis from a project we did funded by the Michael J. Fox Foundation in collaboration with the Mayo Clinic, where we, on the y-axis, you can see previously published data in a mutant model of Parkinson's disease. And on the x-axis is data from our simulation, looking at the same downstream change after a single genetic mutation. And our correlation coefficient of 0.83 is, we're pretty excited about that. So, of course, a new system, and we're continuing to work where we find things that have not been previously published. Of course, have to go back to the lab. This is again where the collaboration with the biologists comes in handy. And we can then say, look, we see these things changing in our simulation. Do you see the same thing in your transgenic models? So now I'm going to walk through a use case and get into some of how we've used this system. So we want to try to understand why Parkinson's happens, where it comes from, how it works, how it progresses. And so to do that, we can look at the causes. We know there are genetic mutations that can cause Parkinson's disease, but we also know that these only account for about 10% of cases. The other 90% are sporadic. But I think it's really interesting to look at the overlap because these two starting points end in the same endpoint pathologies of protein aggregates and progressive cell loss of a particular region. So to look at this, we did 2 manipulations. First, a LRRK2 mutation, where we just replaced LRRK2 protein with a G2019S mutant version. And second, looking at replacing the entire expression profile with that from sporadic Parkinson's patients. So we first run a simulation. We start with a view, kind of looking at an unbiased look at all of the things that change. And so this is, on the y-axis, each row is a biochemical entity within the system. Each column is a simulation time point, and you can see things changing over time. When we then cluster that and do a functional enrichment against gene ontology for the things that change, I'm not going to go through each of these, but there are several biological processes which are implicated by that one thing. Looking at the second manipulation, where we use the transcriptomic data, came from a meta-analysis published a couple of years ago, we see much more diverse changes, and highlighted in yellow are the things that change similarly between the 2 manipulations. So this starts to give us some clues of what might be going on, and we can start to drill in a little bit more. So I'm going to look at one of those things, exocytosis, and we look at the process of synaptic vesicles releasing neurotransmitter, and that's dependent on a protein RAB3, the pink ovals there, being active in order for those vesicles to dock to the membrane to release a neurotransmitter. Well, in both of these models, the active RAB3 levels are lower. So now we can start to put together a hypothesis that can explain why dopamine transmission might be affected. And in fact, in the transgenic animal models, we do see lower levels of extracellular dopamine. So bringing this back to therapeutics, the next thing we do is add a hypothetical drug to the system and see if we can bring the abnormalities back to normal. And this is just one of many of the abnormalities that we measured within this, but we have a target that engagement shows reversal of that inactive RAB3A level. So the next step is molecule development, and so this is kind of our overall pipeline. Our primary business is developing drugs. We want to get something into the clinic, really for the patients. So we have molecules in development at this point. It's kind of an interesting approach rather than high throughput screening and seeing what hits and then going from there to try to figure out why it works or does it work. We start with a validated target and then a rational design for molecules using some of the other computational approaches people here are describing this week.